Hello everyone. I welcome you all to the third lecture of the course Cellular Microenvironment from Oxygen Gradient to Gene Expression. In previous two lecture, we explored the concept of cellular microenvironment, and I also discussed about the dynamic nature of the cellular microenvironment. We also learned how oxygen diffuses from blood vasculatures into the surrounding tissue. So as oxygen diffuses from the blood capillary and it is being consumed by the cells, and its concentration gradually decreases with increasing distance from the blood vessels and resulting in the formation of oxygen gradient within the tissue. But this raises an important question. If oxygen concentration decreases inside the tissue, how does the cell know that oxygen becomes low? Does the cells have a mechanism to detect the oxygen level? More importantly, once a cell sends low oxygen, how does it activate the genes that is required for the survival? So in today's lecture, we are going to answer all of these questions. And, I mean, while understanding the fascinating mechanism of hypoxia sensing through hypoxia incubation factor. So here we will learn how does a cell sense low oxygen condition and how it activates the survival genes for the surviving in that particular microenvironment. And that will be called as the cell's response to the hypoxia. So without wasting our time, let's try to understand first thing here. So in order to understand this, in order to answer the this question, first we need to understand what is HIF1 alpha, and that is the master regulator for the hypoxia genes. And in order to understand the HIF1 alpha, first we should understand what is HIF, right? So HIF is not just oxygen itself. It is, or it is also not an enzyme that produces oxygen. HIF is a transcription factor. That is a special type of protein that regulates the expression of gene. So we can think, transcription factor like a switch inside the nucleus of the cells. So when the switch is turned on, certain genes becomes active, and when switch is turned off, certain genes will become silent. So in that way only, he functions as one of the master switch that allows cells to adapt when oxygen becomes limited. But when we talk about HIF, it is actually made up of two protein subunit, that is a HIF1 alpha, HIF alpha and HIF beta. So first I'll talk about the beta subunit that is present continuously inside the cells and remain relatively stable regardless of the oxygen concentration, and that is called as the constitutively present. The alpha subunit that you can see here, however, it is highly sensitive to the oxygen level. Under normal condition, you will see that if alpha is rapidly destroyed under low oxygen condition, and it becomes stable and get accumulated inside the cells, and it combines with HIF beta to activate the expression of gene that is involved in the survival pathway under hypoxic condition. Okay. So because the alpha subunit, responds directly to the oxygen availability, and it acts as a key regulator for the hypoxia response. So let's try to understand here. This is how this alpha subunit and beta subunit that will go and bind inside the nucleus to the hypoxia responsive element. And in case of hypoxia, as I have told you, that activates the hypoxia responsive gene. So in subsequent lecture, we will also study what is hypoxia responsive gene that gets activated due to this HIF condition or due to this hypoxia. So when we talk about normal oxygen condition, it's very important and very interesting, and this is one of the core concept of this lecture series basically. So in normal condition, that is called as the normoxia. So there will be plenty of oxygen and HIF1 alpha is destroyed. So how HIF1 alpha is destroyed, just try to understand. So let's say you have, inside the cells, if cells feels abundant oxygen. So this PhD is a kind of enzyme that is called as the prolyl hydroxylase domain enzyme, that is called as the PhD. And this PhD enzyme will, hydroxylate this HIF-1 alpha. And what is the hydroxylation? Basically, it will add alcoholic group over the HIF-1 alpha, and that is called as the hydroxylation of HIF-1 alpha. Now, once this HIF-1 alpha get hydroxylated, then VHL is another protein that recognizes hydroxylation alpha. So VHL only recognize the hydroxylated HIF-1 alpha. If it is not getting hydroxylated, then VHL protein cannot recognize this thing. So once this VHL protein recognizes there is a hydroxylated HIF-1 alpha, then what happens? It performs the UV quantization of HIF-1 alpha. And once this process is getting performed, and how this is getting performed with the help of VHL. And again, I'll explain these things, UV quintination, in the subsequent lecture. Here I am just going to let you understand some process or some chemical substitution is happening here. And this UV quintination HIF-1 alpha, enters into the proteasome, and this proteasome basically recognize this UV quintized HIF-1 alpha only. And once it is getting recognized by proteasome, this proteasome basically degrade this HIF-1 alpha unit, right? And that results that the HIF-1 alpha is not getting will not get accumulated inside the cells, and the hypoxia responsive gene will remain switched off. So this is how the pathways of this activation and deactivation of degradation of HIF-1 alpha works when there is abundant oxygen. So in normal condition, in normoxia, you have seen that there is a degradation of HIF-1 alpha, and this degradation is happening due to proteasome basically degrade this UV quantized HIF-1 alpha, and that's why there is the hypoxia responsive gene will be turned off. Now, coming to the second part, when there is a low oxygen, so if cells sense a low oxygen, that is called the hypoxia. So HIF-1 alpha is stabilized, and that will activate the hypoxia responsive gene. So let's try to understand these things here. So when oxygen level decreases, the situation changes completely. Right. So now what happens? This PhD enzyme that require oxygen to function. Otherwise, it will not function. So now, since oxygen concentration got decreased or oxygen is unavailable, the enzyme becomes inactive. Right. And that can no longer modify HIF-1 alpha. So earlier, if this enzyme becomes active, then it will hydroxylate this HIF-1 alpha. But here, due to the limited condition or a lower condition of oxygen, this enzyme will be inactive and it cannot modify HIF-1 alpha. So without this modification, the VHL protein cannot recognize HIF-1 alpha, right? Because VHL, in order to recognize VHL protein to HIF-1 alpha, this HIF-1 alpha should get hydroxylated. Right. So it is no longer, I mean, if it is not getting hydroxylated, then obviously that we have seen that it cannot perform the UV quantization of HIF-1 alpha, and then proteasome cannot recognize this HIF-1 alpha for the degradation. So now HIF-1 alpha will be, stable inside the cells. So instead of being destroyed, HIF-1 alpha begins to accumulate inside the cells, and it then moves into the nucleus. You can see here. Now it entered into the nucleus, and where it binds with the HIF-1 beta subunit, where, as I told you, HIF-1 beta is relatively stable, and that is always present inside the nucleus. And together they form an active HIF transcription factor complex. So this get dimerized, and it basically form transcription factor complex. And now this transcription factor complex binds to the DNA sequence that is called as the hypoxia response element, HREs. Right. This is called as the hypoxia response element, and then it activates the transcription of various genes that helps the cells to adapt the lower oxygen conditions. Right. So now this is how the cells, or this is how this HIF-1 alpha get accumulated inside the cells, that forms the dimer, and once it forms the dimer, that is called as the transcription complex, and then it basically activates the pathway of various genes that is suitable for low oxygen condition. Now, next part is, as I have told you, there are various genes that is being activated by HIF-1 alpha. So here in this slide, we are going to look about the genes that is being activated by HIF-1 alpha. So once it is getting activated, HIF-1 alpha regulates hundreds of the genes, and some of the most important genes includes like VEGF. vascular endothelial growth factor, which promotes the formation of new blood vessels to improve oxygen delivery. So since cells are having various feedback mechanism, and based on that, it will perform this kind of action. Then it will also activates the GLUT1 and GLUT4, which increases glucose uptake so that the cells can produce enough energy for the glycolysis. Then it also activates the EPO, that is called as the erythropoietin, that basically simulates the production of red blood vessels, or sorry, red blood cells, and that enhances the oxygen concentration or oxygen transport. Then there comes a glycolytic enzyme that basically converts this glucose to pyruvate, and finally it produces the adenosine triphosphate ATP, and that basically shifts the metabolic pathway from oxidative phosphorylation to the glycolysis. So, as we have seen this, through these changes, HIF basically helps cells to survive under hypoxic condition. And then there are another things that is also getting activated, like survival pathways, cell proliferation, pH regulation, iron metabolism, and DNA repair, right? So, with that, I would like to end this lecture here only. I hope you'd have understood the concept that if there is a normal oxygen condition, how cells behave. If there is a limited oxygen condition, that is called the hypoxia, how cells behave. So this concept is clear to all of you, and this concept is the heart of this course. So in the next lecture, we will understand that why there are two master regulators for oxygen, like HIP1 alpha and HIP2 alpha. We see that there are two master regulators. So in next lecture, we will answer why there are two master regulators, not only one master regulator for the oxygen limited condition for the cells. Okay. So I'll stop this lecture here. See you in the next lecture. Thank you.